Okay, thank you very much. We just heard what you can do if you once you have a product, how you can optimize the let's say the sellable product, how you can improve the quality of that. What I'm going to do is to talk a bit more about how you get to the product in the first place, and then specifically at a product which has to deal with changing situations of the future. And that has to do with climate change in a broader sense, but more specifically on abiotic stress resistance, because that is where I will be talking about. So this is the outline of the presentation. And I think the reason why we're doing this is because there are quite a lot of threats to global food security. We need to produce much more with less inputs for more people. In the coming years, we get about 2 billion people extra, so we need to produce more food. But at the same time, society is expecting of us to do that in a more environmental friendly way without using pesticides and herbicides and so on. And that is a challenge. It's specifically a challenge because also at the same time our climate is changing, and we have to make sure that we can grow crops which yield sufficiently under all kind of different situations. So we can do that from the management side, try to make sure that we keep everything in control. And of course, if you have a greenhouse, then that is relatively simple. But in the open field, it's much more difficult, especially if the open field is very large. And then you have to make sure that the crops you plant are able to withstand all the problems. So since I'm a geneticist, a breeder, I believe that we can do a lot with trying to improve the crops we are growing from genetics points of view. So we have to make sure that we can produce new crops, and for that we have to make sure also that we can make the crops we know now already better suitable for the growing conditions we want to place them in. But at the same time, we also have to look probably to newer crops, or at least crops which are not familiar to us at the moment, like quinoa, like some other crops which are, let's say, like I said already, less familiar. So I believe breeding is a big solution for that, but not the entire solution. If you look back in the past, then breeding has contributed about 50% to the improvements we see over the last, let's say, 70, 80 years. The other 50% coming from management and fertilizers and a lot of other things. So why deal with abiotic stress? Well, abiotic stress is very important, and of course plants cannot run away once they face stress, so they have to cope with that. Either they try to avoid it, so they try to be outrun drought or be faster in development, or they are tolerant, as I will be talking about, or they try to keep the damage as small as possible. So by dropping the leaves, by making sure that they at least have something which for the plant, from the plant's point of view, is harvestable, ensures that it will face a next generation. Of course, that is not sufficient for the farmers, but from the plant's perspective, that is. Well, there are a lot of abiotic stresses. I've listed here a few: drought or water limitation, flooding, frost, heat, all these kind of things. Of course, also the soils get worse and worse. Here I heard in Australia you have a lot of boron in the soil, to which many plants are not really used. They don't like that so much. But also a lot of salt, of course, and that means that you have to ensure that your crops will be able to grow under these different conditions. Now, there are quite a lot of challenges to trying to breed for abiotic stress resistance or tolerance, and that is because it's quite a complex trait, and that means that a lot of genes are involved. And also when a lot of genes are involved, it means that if you grow in different environments, then the response of those plants might be different. as well. So it means that the ideal situation from many companies is that they design one variety which will be able to grow all over the world and always yield a lot. Well, that is not possible, but you can try to make varieties which do quite well under a whole range of different environments. Now, the trait is complex because many genes are involved, as I already said, and that means that there is a lot of interaction between different pathways in the plant, which in different plants might also be regulated in a different way. So the solution in one plant doesn't automatically mean that that is also the solution in another plant species. If you look to the abiotic stresses or climate change, then you can also see or witness that the different stresses, they vary from year to year. And also they tend to be much more extreme nowadays than they used to be, at least as far as we recollect. And it means that you have to have plants which are able to deal with those very changing situations. So maybe the shift of growing varieties from year to year might be bigger than we have faced so far. Not only because the variety speed or the development of variety is much bigger, but also because we need simply to have other varieties in the different years we are planting. So how can we cope with this in general terms? Well, being a breeder, we want to, let's say, get an idea about what we are talking. So we want to know what exactly the stress is. Because if I ask anyone here in the audience what is drought resistance, then he or she might say this is it. But that's not the only type of drought. Drought, for instance, can be very different when you look at it from a point of view that the drought is inflicted once the plant is in its developmental stage, or whether it is at the harvesting stage. And the effect on the yield will be very different. So what we also want to do is we want to have traits which we can measure, because if we can measure them, then we can maybe design a genomic position or a gene to it, and then we can try to improve and breed for it. Of course, the trait also has to have a good heritability, because if there is only a very small effect from the genetics and a very large effect from the environment, then it becomes difficult to try and introduce this gene in a meaningful way in a new variety. And of course, finally, the trait has to be present in the germplasm, because otherwise we cannot improve for that. That means if you are not using genetic modification. Because if you're using genetic modification, then you could take the traits from another crop and introduce it in your crop of interest. But that is causing a number of other problems, especially with acceptance. So, as I said already, it's difficult to define stress, but it's very important to do so, because otherwise we are not able to really find out which genes are involved, which genes are important. And if you look again at drought, then let's say the severity of the drought period is very important. So the time drought is experienced by the plant, but also the period at which the drought is inflicted. Is it at the beginning, at the establishment of the crop, or is it when the crop is almost ripe? It can also be different whether you have high or low temperatures. So all these kind of things you have to take into account. And you need to know all the different circumstances so that you can really zoom in on the genes of interest for that particular drought tolerance or drought resistance. Well, drought is particularly important, and especially here in Australia, because more than 50% of the arable land is one way or the other affected by drought. And if we look at another important issue, and that is the water usage of crops, then we can see that more than 70% of the fresh water is being used by agriculture. And if we are going to have to feed 2 billion people more, then we will need more water. And then you can question, is there still enough water for people? So if we are able then to grow crops on more salt water or water-containing salt, then that could be quite an achievement, because that would put the pressure or take the pressure off freshwater consumption. So if you look, for instance, here at a potato plant and it experiences drought, then a lot of things will happen. It will see, or it will sense that drought is there, so it will automatically close the stomata, and it will do a lot of other things in order to make sure that the effect of drought on its performance, on its reproduction, is as small as possible. Nevertheless, when it is happening, then you have, in general, two major effects, adverse effects. One is if the drought happens at a certain critical stage before the tubers have been initiated, then you won't get any tubers formed, so you don't have any yield whatsoever. The other thing is that if it is happening at a later stage, then the yield will be dramatically decreased, and your economic yield might not even be there. So it is really important to know when the drought is going to appear or to happen, and what your plant will do in order to respond against it. Well, this is, let's say, a simple one abiotic stress trait, but of course, especially in field-grown crops, there is not one stress at one point. There are multiple stresses, which sometimes also occur at the same time. So how are you going to deal with that? Because that is the real-life situation, not the one stress and then waiting for the next stress to come, and so that it is easy to see what is happening. So we are lucky that we are now in a period where we have a lot of information. We have genomics, we have a lot of tools, and we can now investigate difficult and different stresses at the same time. So the example here is tomato salt stress tolerance at the same time with having a biotic stress, and that is powdery mildew infection. And what we've done is we've done these experiments in greenhouses, because the tomatoes in the Netherlands are grown exclusively in greenhouses. We let the seedlings grow up, and then after about 3 weeks we put them on salt medium, or we add salt to the medium, and then about a week, 10 days later, we make sure that they are being infected with powdery mildew, and then again a couple of weeks later we measure the effect of the powdery mildew infection on the performance of the plants. Now here is a picture of a introgression line population. So this is a number of plants which are offspring plants from a cross between the two plants in orange, as shown here. The one on the far left is the resistant genotype, a wild species, and the one in the middle somewhere, or towards the right, with moneymaker, is a variety. And if you cross those two, you get the plants as shown here, and they respond in a particular way to powdery mildew. But you can see with the light gray bars that as soon as you put these plants on salt and you then apply the powdery mildew stress, that they all seem to be much more susceptible to the disease. And so the higher the value, the higher the susceptibility to the disease. So meaning that salt has a negative effect on the resistance of the different plants. And that's shown again here in these 4 different slides. Here you see the performance of the whole population on 0 millimolars of salt, and they have a value of about, let's say, 2. If you then look at the rest of the population at 50 millimolars of salt, you see that it almost is 1.5 point higher, so being more susceptible for the entire population. But the interesting thing is if you go up further with your salt concentration, then you see that it gets back to, let's say, the normal level where you have no salt. And if you look on how this looks like in real life on the plant, you can see, let's see, there, that the 150 millimolars has indeed less effect of the powdery mildew. So that is interesting to note. Of course, what is still a problem, the salt stress has an effect, and also the disease at the same time, they have an effect on the total performance of the plant. So you have less material, less fresh weight, being quite sure how that looks like in this slide. But it also means that if you have the combined stress, it even has a more profound effect on the total performance of the plant. Now, like I said, we live in an interesting period where we have a lot of possibilities of looking at genes. We have sequences available, and you can, in one particular view, have a look at all the 30,000 genes of the tomato and see which ones are higher expressed or lower expressed, and in that way maybe zoom in on particular genes which are important for giving resistance to this abiotic stress. And that's depicted on this slide. And that helps you then to zoom in on those genes which might be interesting to introgress into the elite varieties to confer resistance against abiotic stress or against the resistance powdery mildew. But the story gets even more complicated if you look at different genotypes. So I just showed you the results from a population, so a cross between two parents, a resistant and a susceptible one. But we also have different mutants which are resistant against powdery mildew. And what you can see on this slide, so you have OL1, OL2, and OL4, three different types of mutants which are resistant against powdery mildew. But where you can see that if you put the OL1 mutant on salt, that it seems to be less resistant. It seems to be more susceptible. So putting that plant on salt immediately results in a lesser level of resistance against the powdery mildew disease. And that is, of course, something you do not want, do not like to have as a grower or breeder. But you can see here the picture at 150 millimolars. I hope you can see it in the back as well. You see more yellow flecks and the development of the disease as opposed to having the 0 millimolars of salt. And here you see the two other mutants, OL2 and OL4, and they are completely unaffected. So it is still possible to have mutants which, let's say, retain their resistance against the disease, but there are also genotypes which lose that resistance. The story even becomes more, let's say, dramatic almost, because this is a nice picture of the OL2 mutant on salt and infected with powdery mildew, and it looks completely clean. There is nothing to be seen, so it is very nice. That is what you like to have. But then if you put the same plant under drought conditions and then infected with powdery mildew, you see the effect on your right, sorry. And there it's quite clear that this mutant is no longer resistant to the powdery mildew. So changing the abiotic stress has a dramatic effect on the resistance against the disease. So concluding from this, thankfully it is now possible to breed for abiotic stress resistance or tolerance, although it is very difficult and complex, but we are now able to do that. It is also clear that the stress level or the stress severity has a profound effect on the actual outcome of the stress interaction in the plant. So that's why it's very important to define your actual stress, which makes it even more complicated because you cannot control drought in the field. Drought in the field is happening when it happens, and yeah, that's something you have to live with. I didn't talk much about it, but it's clear that senescence, so the way of the plant to outrun more or less the stress it's perceiving is very important. Dropping of leaves and so on is very important. And also the stress interaction is also very much dependent on the type of resistance, as I've shown here with the three different mutants. And what is also even more intriguing, but we don't understand yet why that is that some genotypes seem to respond quite differently under different types of stresses. And that's where I would like to stop and acknowledge the people who did the work, most of all Christos Kisudis, the PhD student who did the work on the powder